So it is a pleasure for us to have Professor Wu Li with us today. Wu Li is a professor and director of the IDG McGovern Institute for Brain Research at Beijing Normal University. He's also a director of the State Key Laboratory of Cognitive Neuroscience and Learning. He received his first degree in biophysics from the University of Science and Technology of China, and his PhD in neurobiology from Shanghai Institute of Physiology, the Chinese Academy of Sciences. He did his postdoc, actually it sounds like a few postdocs, with Gerald Westheimer at UC Berkeley, then with Christian Werhahan and Peter Thier at Max Planck Institute for Biological Cybernetics, and then with Charles Gilbert at Rockefeller University. He studies the neural mechanisms underlying visual perception using psychophysical and neurophysiological approaches. And we're very pleased to have you today with us from far away, across, well, it's not exactly across the ocean, but very far away from us, from China. Welcome to our seminar. I want to say to everybody that Wu Li is okay to take questions during the talk. So if you have questions, you're most welcome to unmute yourself and ask questions. And welcome.
Thank you, Sharon. Thanks for inviting me. So today I will share with you our series studies on learning-induced changes in visual cortical processing in awake behaving monkeys. Visual perception requires proper grouping of image components. Image grouping and segmentation follows a set of rules, such as those summarized by Gestalt psychologists. For example, the visual system tends to link counter fragments that showed continuity. Counter integration is a very important step in interpreting visual images. The Gestalt grouping rules reflect a flexible processing mechanism that can be dynamically adjusted according to a global stimulus context. For example, here, the same four lines could be grouped in different manner in different contexts. For example, in this context, it looks different. But in cluttered visual scenes, however, visual grouping simply based on Gestalt rules could be ambiguous or hopeless. In this case, image grouping could be guided by past experience. For example, after a brief exposure to this image, the previous pattern will be reorganized in perception. This could be explained by top-down anticipatory effect based on prior brief experience. Long-term training can also modify our perception, known as perceptual learning. So on the left, we can easily segregate a number 5 from many number 2s. So on the right, the same image is rotated by 90 degrees, but it becomes much more difficult to detect the hidden number 5. So we can, after rotating both patterns, the effect is reversed. So this phenomenon can be explained by a long-term training effect that is specific to familiar shapes. After years of reading experience, we are much more familiar with upright Arabic numerals than rotated ones. So long-term learning and memory comes in many different forms. One form is so-called declarative or explicit, such as remembering some events or facts whose recall can reach our conscious awareness. So my talk today will focus on two forms of implicit or non-declarative learning and memory. One is perceptual learning, and the other is classical conditioning. So for perceptual learning, I will talk about learning to detect task-relevant visual stimulus. I refer to this form of learning as use-dependent because it is effortful and it requires active and repeated engagement in the training task. So for conditioning, I will talk field learning or field conditioning, that is learning to detect threat-associated visual stimuli. So I refer to it as experience dependent because it takes place automatically after repeated pairing of a virus stimulus with an aversive stimulus. Our studies showed that these two forms of learning could induce distinct changes in visual cortical areas as early as in V1. So now let's look at perceptual learning first. So regarding the neural mechanisms underlying perceptual learning, there has been an ongoing debate. So one point of view argues for refined readout process, whereby task-relevant information is read out more efficiently after training. This process could be mediated by the frontal parietal cortical areas. Another point of view argues for refined sensory representation, whereby learning-induced changes reside in the early visual cortex, such as V1. Our study showed that these two mechanisms are not mutually exclusive. So we trained monkeys in a country detection task using a two alternative forced choice task design. So the monkey reported which of these two simultaneous presented stimulus patterns contained a global contour by making a cut to the contour location. So the contour was formed by a number of collinear lines embedded in randomly oriented lines. The contour saliency was manipulated by varying the number of collinear lines forming the contour or the spacing between these collinear lines. So for example, in the left pattern, it's very easy to see the global contour. But in the right pattern, the contour is hidden actually, it's hard to detect. So it basically, you can see if I hide the background. So when the monkey was trained in the contour detection task, we recorded V1 responses using implanted microelectrode arrays. For each electrode or recording site, we measured neurons' responses to the patterns without any contour. We call it noise pattern, and to the pattern containing contours of different saliencies. So the two yellow arrows just highlight the contour path, and the two red circles indicate two V1 neurons with receptive fields lying on the contour path, and two blue squares indicate two neurons on the background. So for each neuron, we measured the distributions of its responses to the noise pattern and to the contour pattern. So we calculated the D prime value as a measure of the neurons' contour detectability. So the D prime could be positive or negative depending on whether the mean neural responses was higher in this case, or even could be lower than that to the noise pattern. So the bottom panels show the data from different training days. These numbers just indicate the animal's behavior performance, which increased with training. This is typical perceptual learning. In each small panels, so red squares just indicate the neurons on the contour path, and squares indicate the neurons on the background. We can see over the training days, there is a progressive increase of neural responses for those neurons on the contour path, and a simultaneous decrease of response of those neurons on the background. So all these symbols are just indicated the center of V1 receptor fields, not the actual size of V1 receptor fields. So we used a support vector machine algorithm to decode the responses of simultaneously recorded neurons. The solid curve indicates the animal's learning curve. 50% was the chance level. And those red dots just indicate the results from SVM classification. So we can see in monkey 1, the SVM decoding performance paralleled the animal's behavior, indicating that the neural representation in V1 was progressively enhanced. And in monkey 2, the SVM decoding accuracy also increased with training. But during the first few days here, so the decoding accuracy was much better than the animal's behavior, suggesting that the animal was unable to fully use or read out the counter signals encoded in V1. After about 2 weeks of training, the two curves converged, so suggesting that with training, the readout process was refined. So note that even after sufficient training, the counter-related signals for both counter facilitation for those neurons on the counter or for background inhibition for those neurons on the background, the counter-related signals are much delayed relative to the visually evoked responses. The early feed-forward component was not modified by training. So the delayed counter-related signals suggest the involvement of feedback influences from higher cortical areas. So in order to dissect the interplay between the feed-forward and feedback processes, so we examined the interactions between V1 and the higher order visual areas V2 and V4 in a series of studies during the counter detection task. So in one study, we implanted microelectrode arrays in V1 and in V4, representing the same visual field locations. So the red symbols just indicate the receptive field centers recorded by the electrode array in V1, and the blue squares those neurons in V4. We simultaneously recorded V1 and V4 neurons in response to contours of different lengths. So the 3 circles here indicate the sample receptor fields recorded by 3 electrodes. So the red circle indicates one V1 neuron with receptor fields on the contour recorded of V1 C site, contour site. So the blue circle of V1 neuron on the background, V1 backgrounds are B site. The large green circle indicates a V4 receptor field, which is much larger than V1 neurons and covering basically the whole stimulus pattern. Neural responses of these 3 groups of neurons are averaged below. So we see that for V1 neurons on the contour, there is response. Different curves actually here indicate different contour lengths. Time 0 indicates stimulus onset, as we can see. The V1 responses for these red guys on the contour increased with contour length. But for those neurons on the background, the contour was running outside their receptive field, and their responses were suppressed by the global contour outside. So,
sorry.
Yes.
May I ask a question? I mean, I want to ask about what is a contour with a length of 1? It's just one item in the receptive field without having I mean, it's just one item?
Yes, with one item in the receptive field surrounded by random oriented lines.
Okay.
In that case, the monkey reported made its choice just by guess.
Okay.
That's why the chance level was 50%. Yeah.
Okay. Thank you.
Okay. So in both V1, both counter facilitation and background suppression. If you look at the PSTH, all these counter-related responses were much delayed relative to visually evoked responses. In contrast, in V4, the facilitatory counter signals emerged at the outset of neural responses, about 40 milliseconds earlier than those V1 counter-related signals. So the visual response latency and the counter signal latency are summarized in this table, and we can see 2 countercurrent processing streams. The visual signals propagate from V1 to V4, but the counter signals propagate backwards. So to examine the feedback influences.
Can I ask? Can I ask a question?
Yep, yep.
Can you go back to the previous slide, please? I didn't get it. If you say, actually, two questions. Basically, the notion is that if you have delayed response, it because top down. But we know, first of all, that the horizontal connection are excitation is slower and maybe can progress. So this is, you can explain it by the same notion that the horizontal connection taking place after 150 milliseconds. This is the first question. Second question, I don't see why the latency of V4 is changing here. Not clear to me about the latency of.
Okay. The first question is very, very, very good, also very important point. I mean, people believe that the speed of horizontal conduction speed of horizontal connection is very slow, relatively much slower than feedback, for example. So here, simply based on latencies, visual latencies, we cannot really conclude. I mean, the delay is entirely introduced by feedback. Okay, so I will give you more data, more evidence later in the following slides, so to show you. I mean,
okay, but I just want to stress that we make calculation of the spread of the horizontal connections.
Yeah.
This is exactly about the time that the feedback come in. It's not
right. Okay. I will. The following slides will answer your first question. The second question, I didn't make myself clear. So, actually, you can measure for each neuron the response latencies, the time required for the signal to be transmitted from the retina to V4, for example. So that's about 59 milliseconds. But if you measure when the counter-related signals emerged in V4, it's about the same time. So that basically there was no delay of the counter signals relative to the visually evoked responses in V4.
But this is because it is the same receptive field. So you have a lot of energy in the receptive field. So it's come very quickly, it's more energy. So it's So receptive field can have the same time the target in the background.
Yeah, exactly. So we speculated that the V4, for example, a V4 neuron can specifically pull some signals from V1 and generate the counter signals in V4. So anyway, I'll give you more data later from data collected in V2, something like that. V2.
Okay.
I go on. Okay. So to examine the feedback influences from V4 to V1, we conducted granular causality analysis on simultaneously recorded spike trains. So we found that V4 to V1 feedback influences were correlated with the counter length for both V1 neurons, the red circle indicate V1 neurons, for V1 neurons on the counter, and also for V1 neurons on the background. So vertical axis just indicated the strength of granular causality, and it's a function in the frequency domain. So different curve indicate different contour lengths. So we can see the granular causality strength is proportional to contour length for both contour facilitation and background inhibition. So this indicates that feedback to V1 is important for both contour facilitation and background suppression. So next, we examined the lateral interactions within V1 between two V1 neurons with different spatial relations relative to the contour. The dashed line just illustrates the contour. So for two V1 neurons on the contour, one on the contour, one on the background, but with opposite information flow, and for two V1 sites on the background. So the red curve in these four panels just results from conventional granular causality analysis. But using conditional granular causality analysis, we can discount or remove the influences from V4. The results are shown as the blue curves. So we can see a general reduction of the granular causality in V1. So using the same conditional granular analysis, we re-examined the feedback from V4 to V1. So here, those dashed curves here. So by removing the influences from V1 background sites, when examining V4 to V1 counter site influence, or removing the influences from V1 counter sites when examining the feedback from V4 to V1 background sites, then we can see a general reduction of granular causality. So this indicates that the feedback and horizontal connections closely interact within V1 to amplify the target counter signal and also to suppress the interfering background noise. So visual cortical areas are interconnected by layer-specific feed-forward and feedback connections. So in another study, we examined the interlaminar interactions between V1 and V2. So we used a pair of so-called linear recording probes, the U probe. So one probe in V1, another probe in V2, all perpendicular. So each probe contained 24 recording sites with 100 micron spacing. So we can target the different layers, different cortical areas. So recordings were made in again in V1, V2 regions, representing the same retinal topic area. Red circles from V1 and blue circles from V2. Just the receptive center again, not the size. Actual size is illustrated here. So for V2 neurons, their receptive fields are on average about 3 to 4 times larger than V1 neurons. So this slide shows the time course of contour signals across cortical layers. So the heat map indicates the proportion of neurons showing significant contour signals across at different cortical depths in V1 and V2, indicated by the y-axis, the relative cortical depths. And at different time points, indicated by the x-axis, time 0 indicates stimulus onset. So the SG indicates the supragranular layer, basically including layers 2 and 3. G, the granular layer, the input layer, layer 4. IG, infragranular layer, layers 5 and 6. So first look at the laminar patterns of the contour signals. In V1, the contour signals are largely confined within the superficial and deep layers, nearly absent in the middle input G layer. These profile of contour patterns are consistent with the feedback, anatomical feedback projections from V2 to V1. So note that in both V1 and V2, the strongest contour signals are present in the superficial layers, the output layers, suggesting that the contour signals are amplified within each cortical area. Now look at the time course. So in V1, the contour signals in all layers were delayed, with latencies lower than 100 milliseconds. For comparison, those small yellow circles just mark the latency of visually evoked responses across different layers. So in V2, which is different from V1, a small proportion of V2 neurons in the superficial layer started encoding the contour in their early response component. In deep layers, middle and deep layers, the contour-related signals are also delayed, similar to V1. So if we average these, we call it early V2 neurons, response from these early neurons. So we can see At the initial burst of neural responses, the contour signals are already correlated with different contour lengths, but these signals are very, very weak. But during the late component, these very weak contour signals are greatly amplified in V2 and also in V1 and as well as in V4 actually during this delayed period. So by placing the global contours at different locations relative to the recorded V1, V2 neurons, we can identify three synergistic processes across V1 and V2. There are three columns here. Column A and B are very similar to the contour facilitation and the background suppression I just mentioned. For suppression, I mean the contour was close to the recorded V1, V2 receptor fields. It's kind of a local suppression near the contour. But in addition to this local inhibitory process, we identified a third, further delayed process by placing the contour in the visual field contralateral to the recorded V1, V2 neurons. So we can see the latency of this third inhibitory process was longer than 200 milliseconds, most likely caused by attention captured by the global contour. So by comparing the laminar patterns, the laminar patterns of these three processes, we can see within the same area, the laminar patterns are very similar in V1, nearly absent in the middle layer in V2. And moreover, for all the three processes, they started actually earlier in V2 than V1. Time zero indicates smooth on that for all three processes. The histograms here just show the V2, V1 latency difference, which are statistically significant actually. So to examine the interlaminar and inter-area interactions and their temporal dynamics, we again used the granular causality analysis within three separate time windows relative to stimulus onset. The first window covers the initial burst of neural responses. During this period of time, the contour signals were absent actually in V1, but very weak in V2 for a small proportion of neurons in superficial layer. During the second time window, the contour signals were rapidly amplified within V1, V2, and also in V4. The third time window, the contour signals reached a plateau in all three cortical areas. So we compared two stimulus conditions here. The contour present, meaning there was a global contour running through the receptor fields, versus contour absent, basically just a noise background. So in each small, it's very busy, I will explain to you. So in each small panel, the y-axis just indicates V1 depths for all of them. V1 y-axis, V1 depths. X-axis indicates the V2 depths. So the left column just results from V1, the feed-forward interaction from V1 to V2. And this column from V2, feedback from V2 to V1. So by comparing the two conditions, actually there are three columns, three rows here just correspond to the three time windows I just mentioned, the early time window, middle time window, and the late time window. So by comparing these two conditions with and without the global contour, we can isolate V1, V2 interactions specific to contour integration. So within the first time window, actually covering the early initial burst of V1, V2 responses, we can see the feed forward and the feedback in these two conditions are very similar. very similar, suggesting that during this early period of time, the bidirectional interactions mainly convey local information, because even without global contour, the patterns are very similar to those with a contour. So within the second time window, when the contour signals were greatly amplified within the cortical circuitry, the inter-area interactions were strongest for when the contour was present, especially the V2 to V1 feedback influences. Within the third time window, when the contour signals are already reached a plateau within the cortical circuitry in V1, in V2, as well in V4, so the inter-area interactions significantly decreased, indicating the completion of the contour integration process. So we further analyzed another stimulus condition when an isolated contour was presented without the background noise. So you can see that bidirectional interactions were mainly confined within the first time window. There was no obvious laminar patterns 100 milliseconds after stimulus onset. This suggests that the contour integration process becomes much faster in the absence of the noise background. That is, there's no it is not necessary to engage the feed-forward and feedback recurrent processing between V1 and V2 to extract isolated contour. So taken together, actually, our results from V1, V2, and V4 recordings are summarized here. So at the beginning of neural responses, local information is conveyed from V1 to V2 and to V4. By pulling the feed-forward inputs, a small proportion of V2 cells in the superficial layer started encoding the contour, into a contour, but the contour signals were very weak, and also for V4 neurons, contour signal also very weak at the beginning. But these early contour signals in higher order areas could serve as a template, and these signals could send back to lower cortical areas, and significantly to amplify the contour signals. And after about 10 or 20 milliseconds delay, the background noise was suppressed. And when the attention, the top-down attention started, actually, we can see the global contour basically across the whole visual field was further suppressed by top-down attention modulation. So further enhance the figure ground contrast signal, making the contour pop out. So here this figure just illustrates the layer-specific interactions between V1 and V2 feed-forward feedback, and the thickness of the area, actually the line into relative stress. So what this is just artwork we just made. We borrowed the so-called thruster effect, to illustrate the recurrent processing among these cortical areas. So basically, the thruster effect means a picture is recursively displayed within itself, resembling our viewing experience between two mirrors facing each other. So through the recurrent processing, actually, all the cortical areas just contained a copy of the contour signals, but with different levels of details. So contour integration is based on global continuity of contour segments. So in another study, we trained monkeys to detect a local discontinuity of a stimulus feature. For example, in the lower left panel, we can easily see an orientation singleton popping out from the background. But if the orientation contrast between the target and the background is small, actually it's very difficult to detect the hidden target. So previous studies have shown that the local orientation contrast is encoded in V1 in a feed-forward manner, just different from the delayed counter signals, global counter signals I just mentioned. So in order to examine whether the feed-forward process could be altered by training, we trained the monkeys to detect the orientation singleton of different orientation contrasts. So here we just use the reaction time task design so that we can measure both detection performance as a proportion correct and also reaction time. So the monkey's behavior increased with training. Different curves indicate different training days: first day, second, third, and remaining days for the hit rate. And for reaction types, we can see progressive reduction for given orientation contrast. We can see a progressive reduction of the reaction types. This is a behavior performance effects of learning on behavior performance. So before training the detection tasks as a baseline control, so we measured V1 responses to orientation singletons of different contrasts. This is population averaged results for different orientation contrasts. We can see the orientation contrast signal is already present at the beginning of neural responses. So this is shown before in previous studies as bottom-up saliency effect. But interestingly, actually, after about 2 weeks of training, if you look at the population response again, so the early feed-forward component was not modified, just as in the global counter detection task. But similar to the counter detection task, the most pronounced changes are seen in the late component of your responses, making the orientation pop-out signals stronger, which corresponds to the improved behavior performance. Even though the early V1 responses were not modified by training, they became more correlated with behavior performance, meaning the readout process is refined. So here, for a given orientation contrast, like in this one, the stimulus condition is the same, but we can separate the collected trials into better trials and worse trials to separate the groups. For worse trials included all the missed trials, and some trials were very long latencies to balance the number of trials in better and worse trial groups. So we can see, even though the physical stimulus was identical, but the neural responses fluctuates. The better trials responses was stronger than in those in the worse trials. So if we calculated the better-worse difference, simply just do the subtraction, we calculate the better-worse difference here, and that if we check at different periods, different time points of learning, first 3 days, intermediate days versus last 3 days, and we can see the better-worse difference actually here increased with training. The last 3 days, the magenta curve here. And the better-worse difference was actually reduced in the late component of V1 responses. For the last 3 days, the magenta curve at the bottom now. So this means, again, as I just mentioned at the beginning, perceptual training can refine both sensory representation and also refine the readout process. So summary of part 1 actually In terms of use-dependent changes associated with effortful and extended practice, training on detection of camouflaged target causes adaptive changes in V1, rendering a more informative representation of the trained stimulus, which in turn contributes to more efficient and less effortful readout at subsequent processing stage. But the changes within V1 are seen only in the late component of neural responses, which are mediated by complex bidirectional interactions among cortical areas. Okay, our perceptual learning studies suggest that the early feed-forward process is rather hardwired. It could not be altered by perceptual learning. However, our recent study showed that the early feed-forward component actually can be altered by fear conditioning. So before the conditioning experiment, we collected baseline control. The monkey was simply doing a simple fixation task. So we measured the neural V1 responses as a function of greeting orientations. This is a typical orientation tuning curve. So the behavioral paradigm of the condition is shown here. The monkey started fixation. So for greetings at a given orientation, for example, tilted leftward, we used it as a conditioned stimulus, which was paired with strong air puff delivered to the face at the end of the trial. And so greetings tilted to the other orientation was used as non-conditioned stimulus. We call it NS, non-conditioned stimulus, as a control actually. So during this period, delay period before delivery of the air puff, we call it a trace interval. We monitored the monkey's eye reflex. So we can see this is the probability of eye closure during this trace interval. We can see for the CS, after presentation of CS greetings tilted, for example, leftward, we can see high probability of eye closure before the delivery of the air puff, but not for the NS condition, just indicating the establishment of the field conditioning. So compared with the pretest baseline control, meaning the monkey was doing fixation without any puff. So we shown as two blue curves here, the baseline control. So we can see in the early phase of V1 responses, there's an enhancement of neural responses only seen in the CS after CS presentation, but not NS presentation. We call it the fear-related signals. So these fear-related signals started very early, with latencies shorter than 40 milliseconds, which is different from the counter signals. So to examine the time course of fear learning in V1, we used a reversal learning paradigm. So we switched the CS and NS orientations in different block of trials back and forth. So this figure are results from two reversals indicated by R1 and R2. So after each reversal, the new CS induced significantly larger fear signals after only a few trials, a few presentation of a few CS presentations, which were randomly mixed within those NS. So about 15 presentations of the CS, we can see the evolvement, the emergence of the fear-related signals associated with the new CS. The second reversal, very similar. So in the post-test, we have a post-test here. In the post-test, the air puff was simply just removed. The CS-NS difference gradually diminished, the so-called fear extinction. So the orientation tuning curves of V1 neurons are also modified by fear conditioning. So we separated V1 cells into 3 groups according to their preferred orientations. For neurons with preferred orientations centered around the CS CS orientation. In this case, the CS was rightward tilted. We can see an elevation of the orientation tuning peak. So for neurons with preferred orientations orthogonal to the CS orientation, we can see an elevation of their tuning flanks. And for the remaining neurons with preferred orientations in between, actually, so we can see an asymmetric elevation. Actually, the CS orientation was on one of their tuning flank. Then in this case, we see an asymmetric elevation of the tuning curve. So the field conditioning can basically rapidly modify the tuning property of V1 neurons. So the field conditioning effect was location specific. For example, if the condition of field learning was established in the upper left visual quadrant, then we introduced the second greeting patch to probe V1 responses. Then we didn't see any field-related signals here. And interestingly, actually, the emergence of field-related signals in V1 seemed to be subliminal. So we superimpose the CS and NS greetings on a horizontally drifting greetings here. So the subjective percept of this compound stimuli is plaid. So in the plaid patterns, actually, the CS and NS orientations were not so clearly visible to the monkeys. But even in this case, actually, the plaid containing the CS greeting, CS orientation, induced very similar field-related signals as CS greetings in V1 early response component. So this further suggests that the field conditioning could modify the bottom-up process.
Can I ask a question?
Yep.
So the conditioning was not done with the plaid. Only after the conditioning was done, you tested it with the plaid, right?
Right, exactly.
Okay, thank you.
We just insert a few trials containing plaids, and you can see the conditioning effect. Yes.
Yeah, thank you.
Summary of part 2. In terms of experience-dependent changes associated with recurring stimulation, V1 neuronal responses can be rapidly modified by field conditioning. The changes in this case are seen from the outset of neural responses, suggesting a bottom-up mechanism that allows for proactive tagging of visual inputs that are predictive of imminent threat. So to wrap up, so our series of studies just indicated that response properties of V1 neurons are pliable, subject to modifications by training and experience. Such flexibility allows more behavior-relevant information to be conveyed at the earliest stage of visual cortical processing. So I would like to thank my students, lab members, and also my collaborators, Zhaoping Li, Lianhua Lou, Charles Gilbert, Peng Guo, and Wu Si, and Marty Rash. Thank you very much.
Thank you very much. And I would like to ask everybody to unmute themselves, and let's give a big applause to Wu Li for a very interesting talk. I'd like to open the stage for questions.
Okay. Well, hi. Very great and impressive work. I really am impressed by the.
Thank you.
How many work? So still going back to my early question or comment, I still think that you can think about V1 neuron properties that you can explain the contour integration already happening in V1 and your learning data. One thing is to take into account, first of all, that you have local inhibition between neurons of the background. So very quickly you can get local inhibiting of the background, which after this, when the horizontal connection or lateral excitation come in, it enhance the contour. I can send you a paper that theoretically, just by properties of V1 neuron, You can see that you can detect without any feedback, you can detect the contour, basically. And with learning, basically, you show that after learning that you get more inhibition on the background, basically, with the excitation. So the signal to noise ratio increase already in V1. So you don't need basically the help of top down. So you can think about it as something that the V1 basically is tagging the location of the contour, so the feedback or attention can shift to the contour location.
So, thank you, Uri. Actually, I think it's worth future studies. We are trying to further dissect the contributions of lateral interactions and the feedback modulations. For example, at the moment, we're also doing some cooling experiment. We're trying to cool down V4, for example, to examine the changes in animals' behavior, also changes in V1 responses. In that case, we can get more insight into the contribution, the feedback, and probably lateral interactions. Thank you. I'll read the paper. Please send it to me. Thank you.
Okay, thank you. Bye-bye.
Can I ask a question?
Sure.
One point I noticed in your data is that the responses in V4 were more sustained compared to V1 that was very transient. Can you comment on that? Why? What do you think is the mechanism?
Which experiment? Counter experiment, right?
In the counter experiment, it seemed that the V4 responses lasted for like 400 milliseconds, while you had a very sharp initial peak in V1 that only later had a contour kind of response. So I wonder whether you can comment, what is the source of sustained response in V4?
Yes, but it depends. Actually, data show here are just mainly from one monkey, but I would say we have tested several monkeys. Most of them just show this kind of sustained pattern with very weak initial burst responses. But here, for all the monkeys, they already are well trained in the detection tasks. Actually, if you look at the pre-training data, actually the sustained activities could be much weaker. You can see clearly an initial burst here. That means, as I just mentioned, probably perceptual learning can induce some pronounced changes in the late component of neural responses, especially particularly for V4 neurons.
Okay, thank you.
Quickly following up on the question of Uri before. So we actually recently showed in mouse visual cortex that that neurons in primary visual cortex actually not just have one receptor field, but a second one, and that the second receptor field, which is spatially distinct and distinct in receptor field properties, is actually driven by feedback. So I think that there is a lot of evidence in the literature out there that these contextual signals are actually driven by feedback, as you're pointing out in your talk.
Yeah, that's a beautiful work. Actually, we shared in our journal club. It's very nice, actually. The feed-forward input and the feedback modulation could have different spatial patterns. There's a spatial range. I completely agree.
Okay, thank you.
I do have a question about the modulation of V1 responses subject to fear conditioning and maybe other emotional modulations. So do you think it has to do with frontal areas or subcortical areas such as the amygdala? And do you know of these connections? Because I'm not aware of them, but what kind of what kind of mechanisms do you think drive these? I mean, I'm sure it's not confined to the visual cortex, but involves
Yeah. Actually, the reviewer asked the same question. Very good question. Ask the same question. At the moment, we don't have any data, but we just speculate because the fear-related signals emerged very early. So, for example, for amygdala, amygdala neurons respond to visual inputs with a latency about more than 100 milliseconds. So that means it's very unlikely that the fear-related signals in V1 simply just somehow came from input from amygdala, in a trial basis. So our speculation is that during the conditioning, over the course of conditioning, the training, especially the early, like I mentioned, something like 15 trials or presentation of the CS is good enough to establish the fear conditioning. So during this period, actually, the amygdala or maybe Pavina could play very important roles in shaping the V1 circuitry to generate these early signals. Because we dig into the literature, there are papers showing that if we silence for example, the Pavina, then the early output of V1, for example, V4 signals will be suppressed. So that happens very early. So we speculated that the origin of the early fear-related signals could be mediated by some subcortical structures. So in future studies, actually, we are going to record from amygdala, from Pavina, to examine their interactions with V1, something like that.
Wow, interesting. So you think it's the connections within the visual hierarchy, but through additional subcortical areas that
right could be doing so, because for both amygdala and Pavina, there are direct anatomical projections to V1.
I see.
Superficial layer, yeah.
Interesting. And you don't think that the frontal eye field is involved because the frontal, I mean, I guess the eyes were not, it wasn't behaving. I mean, the monkey wasn't behaving. The eyes were sleeping.
Right. And also, if the signals are derived from prefrontal area, the latency could be too long.
Okay. Yeah, frontal eye field is about 70 milliseconds. So,
yeah.
Okay, thank you. Okay, so if there are no more questions, I'd like to thank you very much for a very interesting talk. And we hope to hear more about your research in the next, maybe next year. So thanks again for joining us from far away.
Thank you very much.
Thank you.
Okay, thank you, Uri. Thank you, Sharon.
Thank you very much.
Thanks a lot.
Thank you. Bye-bye.
Well, I hope to be able to invite you personally to come here, but you know.
I noticed the dog is there. Hi, dog.
Bye.
Long time no see.
Yeah. So maybe in the perceptual learning meeting this summer, maybe we know.
Okay.